Now, in this session, we are going to discuss about the structure of mammalian nephron. And in the next session, we will start discussing about urine formation, because the process of urine formation takes place in the nephron. So, if you don't understand the structure of the nephron, it will be very difficult for you to understand the process of urine formation. So now, let's go quick to discuss about the structure of mammalian nephron. So each nephron is composed of 6 regions, each having strictly different anatomical features and physiological function. Kwa hiyo about the anatomy, yani structure of the different parts of nephron, different parts of the nephron zinakuwa zina structure tofauti tofauti, na pia different parts of the nephron zinakuwa zina function tofauti tofauti. Kwa anatomy, yani muundo unadetermine physiological function, yani kazi yake. So first we have the renal corpuscle. Sometimes it is called as Malpighian body, which is composed of Bowman's capsule and the glomerulus. Bowman's capsule and the glomerulus. Then we have proximal convoluted tubule, descending limb of loop of Henle, ascending limb of loop of Henle, distal convoluted tubule, and the collecting duct. Now, in this space tutakiwa tu tuna diagram ya nephron, lakini sina diagram ya nephron hapo. But I know you know about the diagram of the nephron. Ukiwa hujui basi we shall see it later. Now, we are starting by discussing about the renal corpuscle. Renal corpuscle or Malpighian body composed of Bowman's capsule and the glomerulus. So, the renal corpuscle of each kidney or nephron has 2 parts, which are glomerulus. And the glomerulus is the network of small blood vessels called the capillaries and the Bowman's capsule, also known as the glomerular capsule. So the Bowman's capsule, which is the double-walled epithelial cup within which glomerulus is contained. So actually, if we want to draw the general structure of the Bowman's capsule, we can say that here we have what we are calling the Bowman's capsule is like a cup. So Bowman's capsule is like a cup, like this way, and here inside is like this way, and then inside of this hapa tutakuwa na blood vessel inaingiza blood, tutakuwa na blood vessel inaingiza blood, na huku tutakuwa na blood vessel inayotoa blood. So na draw it because that is how the anatomy is. So hii blood vessel inside imetengeneza network. Inside imetengeneza network. Kwa hiyo blood ikiingia hapa blood, ndani ikifika ndani inakutana kama na network nyingi ambazo hizi network zinatumika kuifilter hii blood. Zinatumika kufilter hii blood. So ndani ya hii glomerulus ikiingia damu inakutana na network of blood capillaries. Hii network of blood capillaries inasaidia katika kutengeneza resistance, blood flow haraka. Manake hii blood inaingia kwenye network, inazunguka as it passes through the network, materials zinakuwa pumped out kuja kwenye hii hapa space huko. Inakuja kwenye hii Bowman's space. Kwa hii blood inavuka napita huko ndani, materials zinatoka nje. Zikitoka nje, they are collected, and zikishakuwa collected, hii sando huku nephron inaendelea the distal convoluted tube inakuwa iko hapo. Then later we have descending limb of the loop of Henle. Then from there we have the ascending limb of the loop of Henle. And this ascending limb, it has the thin part and the thick part, as how we shall study later. It has the thin and the thick. Then from there hii ina join tena kwenda kuform the proximal convoluted tube. Proximal convoluted tube, then collecting duct kama. Sasa nilichokuwa naelezea ni about the Bowman's capsule. The renal capsule, which is this one. Renal corpuscle, which is sometimes called as Malpighian body. Malpighian body. Renal corpuscle. So it is made up of the glomerulus, which is the network of capillaries, and the Bowman's capsule, which is the cup-like structure. Hizi materials zinakuja huku, and finally this material they will form urine. Kwa kutoka hapa humu katikati ndo kuna different processes zinatokea, tunaziita mechanism of urine formation. So first you need to understand the structure of these parts, and then later ndio tutadiscuss kuhusu hiyo urine formation na mechanism yake, sababu hiyo ni mpaka kwanza umejua tuna structure ya aina gani ambayo tunaita nephron. Now tumeshaona kwamba tuna glomerulus pamoja na Bowman's capsule. So in the renal corpuscle, blood is forced through glomerular capillaries at a high pressure than the pressure to which the blood generally travel around the body, and also into the kidney itself. Kwa hiyo blood inapokuwa inaingia kwenye glomerulus inakuwa inaingia kwa pressure kubwa kuliko pressure ambayo blood inasafiria mwilini. Ikiingia hapo kwa pressure kubwa, if you observe my diagram utaona hii blood vessel ni nene, is thick as compared to this. The lumen of this blood vessel is thicker as compared to this. I mean the lumen of this blood vessel is wider as compared to this. Hii hapa lumen yake ni ndogo ili blood inapokuwa na rotate hapa ndani isinganganie kutoka. The small lumen of this efferent blood vessel, this is called afferent arteriole. This is efferent arteriole. The small lumen of the efferent arteriole help to provide pressure or to provide resistance for the blood in the glomerulus and to resist it from leaving the glomerulus faster before ultrafiltration has occurred. Kwa nachokisema ni kwamba in the renal corpuscle, blood is forced through the glomerular capillaries at a high pressure than the pressure to which the blood generally travels around the body, and also to the kidney itself. Helped by the increased pressure in the glomerular capillaries, a filtration process occurs in which some blood fluid is forced out of the glomerulus and into the corpuscular space of the Bowman's capsule. So, with the high blood pressure in the glomerulus, materials are forced out of blood into the Bowman's space. Some of the blood materials, not all of them, some of them, is how we shall study later in part of ultrafiltration. Now we are saying the fluid that is filtered out of glomerulus into the Bowman's capsule is called the glomerular filtrate. Within the glomerulus are glomerular capillaries that are located between afferent arteriole, bringing blood into the glomerulus, and efferent arteriole, draining blood away from glomerulus. So naona kwamba hii tunaita afferent, hii hapa efferent. Afferent arteriole inaleta blood kwenye glomerulus. Ndani kuna glomerular capillaries, then kuna efferent arteriole. Na utaona kwamba tunasema kwamba the outgoing or the efferent arteriole has a smaller diameter than the incoming or the afferent arteriole. This difference in arterial diameters help to raise the blood pressure in the glomerulus. Kwa hii inasaidia katika kuongeza blood pressure kwenye glomerulus, kama nilivyowaambia kwamba efferent arteriole yenyewe ni ndogo ili kuweza kufanya pressure kwenye glomerulus iongezeke. So about the glomerulus is a knot of blood capillaries. Tunaposema knot ni kama vile kitu fulani ambacho is like a ball. So is like a ball. As if you take this paper, if you take a piece of paper and then you fold it, forming a certain structure. Yeah, this is what you can call as the knot. So glomerulus in drawing tunaichora kama kubwa, lakini it is very small, very small, very small. Size yake ni ndogo sana, So glomerulus is just like a knot of blood capillaries. Ni kama kilivyo hichi kiround of paper, lakini kiwe made up of the blood capillaries, ambacho kina blood inayoingia na inayotoka. So the knot of blood capillaries originating from a single afferent arteriole and rejoined to form efferent arteriole. Actual filtering of blood occurs in the glomerulus. Each glomerulus acts like a sieve that helps to keep normal proteins and cells in your bloodstream and allow waste, excess fluid and other substances to pass out of glomerular capillaries. Kwa hiyo kinachopita kutoka ndani kuja nje, tunakuwa tuna excess materials kama chochote iko excess, then tunakuwa tuna waste product na vitu vingine ambavyo havitakiwi kama excess fluid. Lakini blood cells pamoja na proteins zinabakia kwenye glomerular capillaries na zenyewe zinaendelea na circulation kama kawaida. However, even some of the materials ambazo zina pass kuja huku, they have the chance again to go back into the blood na zikaendelea kutumika mwilini. How? We shall study it later in the process of urine formation. Thus, the glomerulus receives blood from an afferent arteriole, a branch of renal artery. It is thick-walled and has a wide lumen. This is because it carries a large amount of blood at high pressure. Blood is taken away from the glomerulus by afferent arteriole towards the renal vein. This is thick in diameter compared to afferent arteriole, so to increase the pressure of blood in the glomerular capillaries. The glomerulus is enclosed by Bowman's capsule kama ambavyo tulichora kwenye diagram. Here inside you have the glomerulus, then outside you have this is the glomerulus, this is the Bowman's capsule surrounding the glomerulus. Now about the Bowman's capsule, Bowman's capsule. Yeah, about the Bowman's capsule, this is the double-walled capsule or cup. It's like a cup, ni kama kikombe, enclosing the glomerulus, or the round cup-shaped structure that encloses the glomerulus. It is approximately 0.2 millimeter in diameter. So you can see 0.2 millimeter, they are very small. Even this hii tuliyochukulia kwa mfano a knot, hii ni kubwa sana kwa 0.2 millimeter. 0.2 millimeter, they are very small, very small. Kwa hiyo ndio diameter ya glomerulus, I mean the Bowman's capsule. So it is approximately 0.2 millimeter in diameter. It contains a tuft of capillaries called the glomerulus, and the outer wall is continuous with the rest of the nephron. So the space between the two walls of Bowman's capsule is continuous with the lumen of the next part of the nephron. So the Bowman's capsule and the glomerulus together constitute the renal corpuscle or Malpighian body. Kwa hiyo unaona kwamba hii kapu hii umndani mko continuous na huku lumen, na nje mko continuous. So the space of the lumen iko continuous with the lumen of the nephron, and the outside iko continuous with the outside wall of the nephron. So nachokisema ni kwamba the entire structure of the nephron corpuscle is related to this function of filtering blood. How? Unaweza kuulizwa kuhusu adaptation kwamba kivipi, how the renal corpuscle is adapted to this function of filtering blood. Renal corpuscle only, sio nephron yote, renal corpuscle. How it is adapted to this function of filtering blood. So firstly, the epithelial lining of the Bowman's corpuscle has a minute pores that allow passage of small molecules such as sugar, salt, urea, and water molecule. However, the large molecules such as proteins and blood cell are restricted from passing through the pores. Kama tulivyosema kule mwanzoni kwamba sio materials zote zinazoweza kupita kwenye hiyo renal corpuscle. Kwa nini? Kwa sababu materials nyingine zenyewe zinakuwa restricted kwa namna ile pale, ili kuweza kufanya vitu viende kwa namna ambayo inatakiwa. Then the second adaptation of the renal corpuscle, the epithelium of the renal corpuscle or Bowman's capsule is made up of special cells called the podocytes. Hizi zote tutaenda kuziona kwa ukaribu zaidi. Podocytes, which together with the neighboring cell form slit pores or filtering pores for ultrafiltration. Kwa kwanza tumeona kwamba epithelial lining ina small pores, lakini sasa tumeona pia epithelial cell ina special epithelial cell ambazo zinaitwa podocytes. Lakini tatu, the basement membrane of the blood capillaries contains several fibers including collagen fiber. These play key role in preventing the passage of protein and blood cell which has large size. On other hand, the charges of fiber repel the negative charged proteins, thus restricting them from passing through the pores. Kwa kuna collagen fiber ambazo zenyewe ziko negatively charged. Sasa negatively charged collagen fiber maana yake protein nazo negatively charged. So they repel each other. Na hiyo ni moja kati ya mechanism ambayo protein haziwi filtered kwenye kidney. Kwa sababu sio matobo yote ya kwenye epithelial lining ni madogo. Matobo mengine ni makubwa. Ila the second mechanism ambayo inatumika huwa ni hiyo repulsion of the negative charges of the collagen, that of protein. So the capillary walls of glomerulus are composed of a single layer of epithelial cells with opening out tunazita fenestrations 50 to 100 micrometer of diameter to restrict penetration of blood cell and proteins. However, as I told you, there are some of the proteins which are smaller than even 100 micrometer. For example, they are 90 micrometer in diameter. So why are they not crossing the capillary wall? It is because of the repulsion caused by the collagen proteins. Kwa hiyo ni adaptation ya tatu. Adaptation ya nne, the endothelial cell of capillary wall are pressed up against the basement membrane au basal lamina, which completely envelopes each capillary and forms the only continuous structure separating the blood in the capillary from the lumen of the Bowman's capsule. Hiyo ni adaptation. And adaptation ya tano, the basement membrane contain collagen for structural purposes and negative charged glycoproteins that repel only negative charged albumin and other protein. Hii tunaita electrophysiology. This adaptation is somehow similar to the third one, but this is strictly explaining about the electrophysiology. Then another adaptation, the inner layer of Bowman's capsule is composed of cells called the podocytes. Extending from each podocyte are thousands of foot-like structures called the pedicles. The space between pedicles are called slit pores or filtration units. Thus, facilitate the process of filtration. Therefore, fluid filtered from blood into the lumen of Bowman's capsule must cross in a close layer tatu. 1. Capillary wall au naita endothelium of the glomerulus. 2. The basement membrane of the capillary. Na 3. The inner visceral layer of the Bowman's capsule au the epithelial lining of the Bowman's capsule. Kwa hiyo hapa patakapo diagram ya hiyo renal corpuscle, ila hiyo diagram haijakuwepo hapa. Ngoja nimalizie na structure nyingine za renal corpuscle za nephrone hapa. Tumalizie hapa faster then tuende kwenye uniformation. So tuna proximal convoluted tube is the longest and highly coiled part of the nephron and conveys filtrate from Bowman's capsule to the loop of Henle. It is 14 mm long and 16 micrometer wide. The proximal convoluted tube is composed of a single layer of epithelial cell with extensive microville forming a blush border on the surface lining the tubule. So the inner epithelial cell pamoja na microville. Microville zinafanya kazi gani hapo utaona mbele kwenye urine information. The tubule has numerous mitochondria concentrated near the basement membrane where they provide ATP for membrane-bound carrier molecule involving in the active transport during reabsorption. Active transport during reabsorption is one among the stages for urine formation. It is the site for selective reabsorption in which the useful materials such as sugar, some salts, and useful amino acids are extracted from renal fluid and transferred back into the bloodstream. So it starts from the neck of the Bowman's capsule and is highly convoluted. Highly convoluted, that means it is highly coiled. It lies in the renal cortex. The wall of the proximal convoluted tube consists of single layer of columnar cell bearing a lot of mitochondria and microville in the surface to increase surface area for selective reabsorption. Number 3 is descending limb of the loop of Henle. We have no more explanation about this, but we'll discuss it later in deep when we'll be discussing about the countercurrent multiplier. So the tubule is permeable to water, thus water moves passively out of the filtrate, that means urine, by osmosis. It is permeable to water. At the same time, there is some passive movement of chloride and sodium ions into the descending limb. So out. Hence, resulting in the progressive increase in the salt concentration at the urine filter moves down through the loop. Kwa hiyo inavyotoka juu kushuka chini, concentration ya urine inakuwa inaongezeka kwa sababu maji yanakuwa yanatoka. Number 4 is about ascending limb of the loop of Henle. Here, chloride and sodium ions are actively transported out of the nephron. Out. Actively transported, maana hii ni active transport inatumia energy. Out of the nephron into the surrounding tissue, and the tubule is impermeable to water. So the urine becomes dilute as it ascends this portion of the nephron. It becomes dilute. Note, the amount of sodium salt in the blood is controlled by aldosterone. Hormone produced by adrenal glands. So the release of hormone leads to the more sodium ions being reabsorbed from filtrate in this part of ascending limb of loop of Henle. From there we have the distal convoluted tube. Here, a controlled amount of water is reabsorbed into the blood by osmosis. Low amount of water in the blood stimulates the pituitary gland, which secretes antidiuretic hormone, ADH, or sometimes it is called as vasopressin, to make the wall of this tube to become more permeable to water. Hii tenda kuona kwenye osmoregulation, maintenance of water, osmoregulation. Kwa hiyo hapo ndiko ambapo ADH huenda kufanya kazi. And finally, we have the collecting duct. Each collecting duct serves several nephrons draining the urine into the pelvis of the kidney. So the final adjustment of urine composition occurs in the distal convoluted tube. After there, no further changes before the urine is removed from the organism. So, collecting duct kazi yake ni correct to urine na kutoa nje. Note, at one point of the distal convoluted tube is joined to the afferent arteriole. Distal convoluted tube is joined to the afferent arteriole by means of a muscle of glandular structure called the juxtaglomerular complex. Some of people they call it as the juxta. I call juxtaglomerular complex or juxtaglomerular apparatus, which consists of the juxtaglomerular cells or the juxtaglomerular cells and the macula densa cells. So what are the function of the juxtaglomerular cells and the macula densa cells? Tenda to discuss later. This play key role in osmoregulation. Hii ndio structure nephron. Hapa tumekuonyesha distal convoluted tube ime contact pale na juxtaglomerular complex. So In this part you may be asked the question with the aid of a diagram, explain the structure of nephron. Lakini mimi nilikuwa nakuelezea ili tuelewane process ya urine formation. Now tunaenda kwenye urine formation.